This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 257, recorded on November 1st, 2013. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today here in the TWiV studio, Dickson Despommier.
Hello, Vincent.
How are you, Dickson?
Chipper. Feeling very good today.
Are you chopping wood?
Not in the Fargo sense of the word. No, that's a different kind of chipper.
So you're feeling good today?
I feel great.
What's the weather there? Can you see out the window?
I can see out the window. It's partly cloudy. It's cold. It's windy. It's the temperature's down around 48 degrees, I would guess.
No, it's not.
It's above that. It's above that. That's right. It's going to be that tomorrow.
Bloody 20 degrees C today.
Yeah, it's going to be around 70 with winds. We had a very interesting rain event this morning on the way in. I know you got caught on it. I didn't.
Well, actually, we had lots of wind.
We did.
Power line got knocked down.
Really?
Right next to my kids' high school.
No kidding.
Caught fire.
Gadzooks.
So all the kids were evacuated.
Yow.
And standing in the rain.
Oh, my.
And it was quite an interesting morning.
Wow.
Also joining us today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody.
Oh, Kathy.
We had a wind advisory this morning, too. It was overnight and this morning.
Right.
And it is 48 degrees here.
We're going to get your weather tomorrow.
Yeah, it's 9 Celsius.
It's 20 degrees.
Sorry about that.
Yeah, it's interesting. It's chilly out there.
I agree.
Well, we had a cold spell, and now it's getting warm again.
Right.
It's weird.
And tomorrow will be cold.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
Alan.
Probably have our weather,
we had
right?
the same snotty weather as you did. It blasted through here. They were saying wind gusts up to 40 or 50 miles an hour, and pouring rain, and then stopping, and then spitting rain, and
it's
really blustering.
It's the 1-year anniversary of Sandy.
That's right.
Oh yeah,
the remnants of Sandy,
and approximately the 2-year anniversary of the October snowstorm that knocked out my power for a week.
Yes,
I remember that.
Yeah, annihilated a whole section of New England. It just blew everything down.
Amazing.
Actually pulled everything down.
Yeah.
Also joining us today from North Central Florida, Rich Condit.
Hi, everybody.
Hey, Richard.
Hey, Rich.
How you doing?
Yeah, what's like down there?
We got 82 Fahrenheit,
cool,
which is 28 Celsius. It's mostly cloudy, which, I find unacceptable. But I just have to deal with it. 60% humidity, dew point 67 degrees, wind out of the southwest at 12 miles an hour, gusting to 19. So it could be better, but I'm not complaining about the temperature.
It's probably perfect Burmese python weather, though.
Oh, yeah. That's probably true.
Humidity, is that it, Dickson? Humidity?
Humidity, that's right.
Humidity,
that's right.
All right. The weather having been set.
Right.
Now we can proceed. We have a couple of papers today. We have one science paper and one non-science paper, but about science.
Philosophy paper.
Philosophy, perhaps.
The PH part of it.
Yeah, right. But before that, we have a couple of items of business. The first comes from an email from Patrick, our friend out in the Malik lab at the Hutch. He writes, Dear TWiV team, I'm writing with the sad news that Leonard Herzenberg passed away this morning, Sunday, October 27th, 2013. Len was a brilliant scientist, and among many major contributions to the field of immunology with his wife and colleague Leonore Herzenberg, he is perhaps best known as the father of flow cytometry. I've attached a short autobiographical sketch that was published a few years ago outlining their partnership and shared contributions to science. Len was also a fantastic human being. Together, Lee and Len have built a legacy not only through their research, but also through countless trainees that are very much part of the Herzenberg family. Lee and Len took a chance on me as an undergraduate summer research student, and I'm ever indebted to them for giving me my start in science. Thank you for sharing their story. So he sent a link to an article from the annual review of Immunology, which is an article written by, it's sort of a biography or autobiography of the two of them, written in a really nice style. It's a lot of fun to read. Mm-hmm. It starts way at the beginning. Well, not
before
when
they were haploid.
No, it starts when they were just before they were married and students and so forth. But it's nice.
It's a great story.
Link to that. It's really cool. So thank you for that. I knew their names for many years because of the facts. The flow cytometry machine. They basically kept reinventing it all the time, making it better and better.
Nice.
Very big contribution.
Huge.
I also want to point out I've just today posted a very nice collection of reminiscences about John Holland over at virology.ws. Many thanks to Kathy for the photo, which I think is pretty cool. Mm-hmm. He used to wear those, what do you call those kind of string ties?
String tie. That's the only time I ever saw him wear that.
Cool.
No one. or of any tie of any sort, so it's sort of not in character, but it was for the symposium held in honor of him when he retired, so that's
it's like if I absolutely have to wear a tie, this is going to be it.
That's right.
The minimum tie.
Yep.
It's usually a Texas thing.
The blog is really nice, so I think you got a good cross-section of things that some things I'd forgotten about, some things were exactly what happened to me too. So
I asked Bert Semler to just sum up his history, which he did. And then I asked a number of people, former trainees, whose emails I got from Kathy. And all but one responded. And they're all here. And they're all a lot of things overlapping, which is really interesting. And a lot of interesting stuff. So check it out. That's pretty neat. And I hope everybody enjoys it. His former people should enjoy it, I would guess, right?
Yeah, and other people who just want to hear about different mentoring styles. Yeah. And I wanted to follow up on two things. In the video, which is his lecture, he talks about ticks being insects. Oops. And we would have really womped on him for that. And that kind of came out in someone's reminiscence that there wasn't any topic that he didn't know something about. But when we found something, we just were relentless. And then the other thing I wanted to mention, when I listened to last week's episode, when I said that Isabel was from Toledo, I should have said Toledo, Ohio.
Right.
And if I had meant Spain, I would have said Toledo. So
there you go.
We'll probably do that. But,
That's right.
Right. But they do have something in common, by the way, because they do talk with each other. Those two cities do that.
Is that right?
I once lived in Toledo, not Toledo. Toledo. They call it Toledo out in Ohio, not Toledo.
All right. And now on to our paper. It's a Nature Immunology paper. We don't often do papers from that journal.
No.
It's what paper that Kathy. Well, you sent me a science summary. Where was the original summary?
It's from the Scientist.
The Scientist.
That's a nice
summary, and this is the original paper. It's called "The Kinase mTOR Modulates the Antibody Response to Provide Cross-Protective Immunity to Lethal Infection with Influenza Virus." It's from a group of individuals at St. Jude's, Fred Hutchinson, and also St. Jude's. And the authors are Keating, Hertz, Wenkel, Harris, Edwards, McLaren, Brown, Sermon, Wilson, Bradley, Hurwitz, G, Doherty, Thomas, and McGargle. I thought this is a really cool paper. And I just listened to this paper in light of the next discussion we're going to have. All right?
Yeah.
Just keep that in mind.
Yeah.
Yes.
So before we do the science, I need to vent a little here about the introduction to this paper.
Oh dear.
All right. Does anyone object?
No.
No, go ahead.
I've never heard you vent.
No.
So in what is becoming more typical fashion, they spend 5 or 6 paragraphs justifying this work in terms of human disease, which is not bad, but there's still science here, okay? So they start out by telling us how flu kills a lot of people, and then they tell us that the mortality rate for H5N1 is 59%. All right? It's not the mortality rate.
Correct.
It's the case fatality ratio.
Here.
Well, they do say for the 637 humans confirmed as being infected.
It's not the mortality rate. That is a specific term.
It is a term of art that is misused.
It's the case-fatality ratio.
Yeah.
Right.
Next, ferrets are generally considered a surrogate for spreading among humans. No animal is a surrogate. They are animal models. They have,
and ferrets don't spread among humans all that often.
They don't. You could ferret out an idea, however.
All right. So then they start to talk about vaccines. They say attempts to generate broadly protective vaccines have had very limited success thus far. And they mention that mainly these are using internal epitopes to the virus. Now, I would disagree with that statement. I think vaccine broadly neutralizing antibody attempts have been pretty good in animal models so far, using either stem antigens or other approaches. So I think that's not a fair summary of the literature. Okay. All right. So those are some of my main objections. I think these are really glaring. I don't know why they're doing this, but I don't mean to take away from the science. But all right, those are my issues.
Reminds me of a conversation I had early on with my graduate mentor, who told me that, there's basically two parts of a paper. There are the experiments and the data, and then there's what they say about it. And no matter what they say about it, the experiments and the data are there. Right. You can throw away everything they say about it. And still, if you don't like what they say about it, the data are still there for you to consider.
Yeah.
Sure. But I find this a trend now where more and more you see all this stuff up front, where if you go back 30 years to a virology paper, they'll go, first sentence, we were interested in knowing how many proteins there are in the core of VSV or something. To do this, we ran sucrose gradients. You get right into it.
And what were the grant pay lines then and now?
Yeah, well, I think that's part of it. That money is tight.
That's a huge part of it.
I think the public is also part of it, and the journals want an important thing. Blah blah blah.
This issue dribbles over in a big way to grant writing. And I don't know how you guys feel about it, but frankly, for me, if I have to wade through, a couple of pages of, sort of hyped up justification in order to get to what the question is, I'm already annoyed by the time I get to the question. Okay?
Exactly.
So spare me all the hype and tell me what the question is.
I mean, this is not to take the science in this paper is very good, and I think this detracts from it in a way. You can ignore it, as Rich says, but I find it annoying and unnecessary to say these things, especially to say incorrect things. To tell me that H5N1 is going to spread among humans because of a ferret experiment and that 60% of the people die, it's just wrong. You don't need to do that. We know we need better flu vaccines.
Right.
All right.
Okay.
The central player in this paper is a protein complex we have never mentioned on TWiV. It's called mTOR. Little m, big T, big O, big R. And it's mammalian target of rapamycin. All right. It's a complex of a number of different proteins. And rapamycin is actually a product originally obtained from Streptomyces. It was first found as an antifungal, but it turns out to be even more important. It's an immunosuppressive drug. Mm-hmm. It's often
in fact, it didn't work out as an antifungal, as I recall.
And
my guess is it's too the side
immunosuppressive.
Yeah, because it's immunosuppressive.
So it's used clinically in people organ transplant. I was just talking to my colleague Chris Schindler this morning. He says when people have autoimmunity issues, they use it. They use it in transplant. And its target, as you might guess, is mTOR. And
Do you know why rapamycin is called rapamycin?
No.
Because the strain of strep that it was isolated from was found in a soil sample on Easter Island, or Rapa Nui.
Cool.
Wow.
Nice. I like that.
Wow.
You're going to build that into the title, Alan?
We could.
We already got a title here.
Yes, we have a great title. All right, so one of the things that rapamycin does is to mess with lymphocytes. It enhances the production of memory CD8 T cells. So the CD8 T cells are the cytotoxic T cells, so this enhances the memory of them. As an immunosuppressive drug, it blocks proliferation of B and T cells. It also increases. I said about the memory CD8 cells. So they wanted to know if this drug would have any influence on influenza and generation of immunity to it. And that's really basically what we're starting from here.
So mTOR is central to this whole convoluted pathway that governs which way the immune response is going to go.
Yeah.
And rapamycin has this fairly distinct effect on it.
So mTOR has a number of interesting effects, and one of them, it is basically a sensor of nutrients outside the cell. It's one of its roles. So it's downstream of a cell surface sensor, and if there are plenty of nutrients, mTOR will basically say to the cell, you can make all the protein you want. And if there's not a lot of nutrients, it will close down protein synthesis. But it also has a role in lymphocyte maturation, and it does it in antibody diversity, as we'll see, and it does that in various other ways. So the model they use is mice, which is not a great model for flu, but it's a great model for immunity. Mm-hmm. They infect mice intraperitoneally, and then they challenge them intranasally. And the virus that they infect with them IP is it's actually an old vaccine strain called X31. It's an H3N2 influenza virus. This was a vaccine used during the 60s. I remember everyone was always talking about X31.
Could you just briefly explain to those not terribly familiar with what represents a good animal model for influenza, why mice are not a good animal model?
People are a great animal.
No, I know that, but tell us why. Tell us why. John Steinbeck
Well,
knew
first
that.
of all, most strains of influenza virus do not replicate in mice.
Right.
The most commonly used strain, which we've talked about here on TWiV, is PR8, which was isolated in the 30s. It's an H1N1 strain, and that was adapted to mice, and so people are very suspicious of that. And so you don't get a full replication. The mice don't get, influenza. They can lose weight and they can die with certain strains, but it's not really recapitulating.
And you know exactly why that is the case?
Do I know why they die?
Yeah. Or why they are not a good model for the wild strains that infect people.
Well,
what is the molecular reason?
Oh.
No, there isn't a single molecular reason. No, there are many. There are probably many reasons. One of which most of the viruses don't replicate, but when they do, they don't do the right things. They go to the CNS, or they do other things that aren't related to flu.
Okay. I just as a curious.
Yeah, no, it's fine. It's a good question.
And depending on how you want to slant it, the mouse immune system has some significant differences from the human immune system.
Absolutely right.
Sure.
And the mouse immune system has some significant similarities.
I mean, as we'll see in the end here, they make some interesting observations, but they will have to be validated in other models and eventually in people.
Right.
But it's very it's the way
One thing
animal.
that does seem to be conserved between people and mice is this response to rapamycin, though, right?
Aha.
Apparently, yes.
So you see this enhanced memory T cell CD8 T cell effect in mice, and in fact, there's some prior work on this where people infected mice and gave them rapamycin to see what would happen with other pathogens.
That's actually an interesting point, Alan, because my sense, not that I have deep insight into this topic, but my sense is that they're uncovering some new stuff about rapamycin in this paper.
Yeah.
Yes.
And it will be interesting to see if that actually holds up in humans.
That's right.
Which is why you do animal model experiments, because you can dissect these mechanisms and figure out what's going on.
So, someone who's just got an organ transplant, do they have to stay on rapamycin for the rest of their life, like cyclosporin A people do?
Don't know.
Because if they did, then that would be the experiment to look at.
I'm a scientist, not a doctor.
Well, actually, Dickson, we're going to get to why that's not quite the natural experiment that you want. Is because what you're getting at is, can't you just look at incidence of flu in people who were organ transplant recipients, and even if they were just purely on rapamycin, that probably wouldn't get at this same mechanism.
Right.
So the way this works is this virus, X31, they write, when given intraperitoneally, it undergoes a defective growth cycle and produces the full spectrum of viral proteins but no infectious virus. And I read, quote, similar to a vaccine, unquote. Now, that's not quite right because the one of the flu vaccines, of course, is inactivated and doesn't produce the full spectrum of viral proteins.
Right.
I mean, it can produce it, but in the case of the flu vaccine, it's either infectious or inactivated, so that's not really quite the same.
The infectious flu vaccine is a little bit different from this one.
That's right. Nevertheless, they're sort of immunizing these mice with intraperitoneally, and then they treat them for 28 days with with rapamycin or without rapamycin, and then they challenge them intranasally. So the idea is, will rapamycin influence the memory response or the challenge to a different strain? And they're using an H5N1 challenge strain here, so they're looking at a different subtype. So,
and in supplemental figure 1, you see that the mice are quite happy about this.
About what?
this whole experiment. They have these very cartoonish, smiling mice.
So they use an H5N1 that's been modified so they could use it at BSL2 conditions. They've replaced all the internal genes. They've replaced the polybasic cleavage site in the hemagglutinin, which all of this allows you to use it at BSL2, which is a good idea so that you don't need to deal with BSL3+. So what they find is when you do this, you immunize IP, give them rapamycin, challenge with H5. With rapamycin, the mice live longer. More mice live. They lose weight, but they gain it back. They lose less weight. They're clearing virus faster than the PBS control mice.
In fact, in the initial experiment, all the rapamycin
they all survived.
Lived, and the ones that got the buffer are croaking, and I think only 40% or 30% of them survive.
So it's quite it is a clear difference.
Right. So just to emphasize this point, this is pretty dramatic because remember the background here is we have to change flu vaccines every year because of antigenic drift in the influenza strains, and in fact, that's antigenic drift even within a given subtype like H1N1, you can get antigenic drift to a different H1N1 and have to make a new influenza. And here we're talking about what is effectively antigenic shift. It's two very different flu strains, and the immunization against one of them in the presence of rapamycin is giving cross-protection against a very different flu strain. So in terms of the ultimate goal of making a universal flu vaccine, this looks on the surface terrific.
Right. They also... they did this second experiment. All right. I've just blanked on this.
I just erased your head, right? Sorry.
No, you didn't do anything. They treated mice with rapamycin, then they infected them with a temperature-sensitive strain of virus, then continued. So in other words, they didn't do IP with an inactive, non-replicating virus. They infected them intranasally with a virus that replicated, treated them with rapamycin, and then challenged them with the H5, and they basically get similar survival and effects on weight loss.
Well, wait, didn't they give the virus and then the rapamycin?
Yeah, they give virus well, that's the same as before. They immunize, they give rapamycin.
Don't they later put them on rapamycin and then give virus?
That's later.
Right, okay.
Right, this is just this other little experiment to make sure that IP isn't, there's nothing special about IP.
Nothing special about IP, but they're still giving virus and then rapamycin.
Yeah, they do IN with a replicating virus, then they give rapamycin, then they challenge, and they get the same effect. Okay.
Right.
They also, yes, go ahead.
I was just going to say one thing. The Scientist article had a beef with the fact that they use these temperature-sensitive viruses, or that they weren't enough like vaccine viruses. But in a sense, doing this temperature-sensitive thing is somewhat like the flu mist.
Yes.
Yeah, it is. Yeah.
So
it's very much like flu mist, but
that's right. That's right. In fact, that's what they're trying to mimic here. Yeah. They show that this kind of protocol, IP rapamycin challenge, will also protect against H7N9 and H1N1. All right, so remember we're immunizing with H3N2. So
Not quite as well.
Not as well, but, you know,
Still works.
Still works, and you can imagine tweaking and getting it better.
Suggest that there's a common antigen here someplace, yes?
Maybe, Dickson. We'll see. Keep that thought in mind, okay?
Yes, good thought.
Hold that very small thought.
You need that first IP inoculation for this to work. If you just give mice rapamycin and then challenge them, you know, they're not protected.
Right.
Which is good.
Yep.
If you do this protocol, it doesn't protect you against Sendai virus.
Nope.
Which is good. So you're not just getting some general, I don't know, immune response. And then they start to look at why this is working. Because, you know, we said before that rapamycin is playing with T cells. So let's look at T cells. So they measure virus-specific T cells after infecting mice and treating them with rapamycin or PBS. And they see that rapamycin indeed increases the number of memory CD8 cells, and you can measure them in very specific ways that we don't need to go into, but they're really cool and really high-tech.
Yep.
Right? So you get an increase in CD8 memory T cells. So then they ask, do you need these T cells? Are they part of the effect? So they do two cool experiments. One, they remove the A T cell epitope from influenza virus. So there are 5 known dominant CD8 T cell epitopes in the virion. So they take out one, and they use this to infect mice. And they show that, in fact, the mice don't make CD8s against that epitope, which you would expect. But you still get the rapamycin effect. So it doesn't matter if the CD8 epitope is gone. I want to say CD8 episode, right? And then they actually ablate CD8 cells. They deplete them with antibodies to the CD8 protein, and they still get the rapamycin effect. So you don't need CD8 cells.
Has nothing to do with the CD8 cells.
In fact, we're talking about memory CD8 cells specifically. It doesn't have to do with what they thought rapamycin was doing in the first place. Isn't that,
so rapamycin does what what people have said it does. It increases the memory CD8 cells, but that's irrelevant.
Right.
That's irrelevant to this effect.
Correct.
To this effect.
Yeah.
So Dickson, if you did these experiments and you found CD8 cells were irrelevant, what would you do next?
Quit.
You quit? That's it? Go home?
Go home.
Let's see about the CD4 cells.
I'm glad you said that. I'm glad you said that, because that's
I would divide and conquer.
That's the next experiment. They deplete CD4 positive T cells. Do the same protocol.
Right.
And in this case,
you need one of the two. Those you don't have anything left after that.
They need CD4 cells.
Yeah.
All right.
Yeah.
Good thing, because if that hadn't worked, it would have been time.
You need them.
Right.
You need the CD4s. They do another experiment where they ask, do you need them during the first infection or the second infection? And the CD4s need to be there during the intraperitoneally administration of the X31 virus.
Roger that.
You know, later on it doesn't matter. So that kind of, CD4s are involved in what, Dickson? Do you remember?
Antibody formation.
Yeah,
and stuff
they
like
make cytokines that help mature B cells, right?
Yeah.
So maybe there's some antibody thing going on here, right?
Yeah. Do I remember?
Do you remember?
I live that.
You're always
day.
saying that you don't know anything. So
this is true.
I'm
This
just
is true.
showing that you do, in fact.
No, don't go any further. Just stop right there.
That's as far as I can go?
Stop.
Okay. They also do some timing experiments here.
Yep.
And they give them rapamycin for different periods, and they find that the most crucial is the first 15 to 20 days after infection with the intraperitoneal infection with X31. So again, it's consistent with, making antibodies, right?
Yeah.
A lot of other things too.
Right, Vincent.
And that gets back to your question about transplant patients. If you had somebody who's on rapamycin long term, you wouldn't be able to get this effect.
Right.
If this is even going on in humans.
If this is even going on.
You got to have it around when you're some early period, a couple of weeks behind when you give the antigen, right?
Right. It's got to show up right after you get the antigen.
You got it.
Sometime after you get the antigen.
Okay.
They rule out regulatory T cells as having an effect here.
Wow.
Okay. And so now they say, okay, because CD4 cells are involved, and as I said before, Dickson, and you so aptly responded,
Yes.
Antibody. What makes antibody, Dickson?
B cells.
Exactly. So let's see. There's a way to make mice without B cells.
He's
Dickson.
showing me cue cards, by the way, everybody. I know you can't see this, but he's got these gigantic handwritten cue cards, erasable, and he's got all the answers, so I'm actually cheating my way through this podcast.
I'm just trying to show that you actually know stuff and you're a scientist because you can figure stuff out. The details are not important. So you can make mice lacking B cells. Okay,
Right.
You can do a genetic ablation. They're called mu-MT mice, and they don't make B cells.
Tough to stay alive under those conditions.
Yeah, if you don't, you grow them under the. You grow them.
You have to plant them shallow, not too
You
deep.
use sterile techniques.
Sterile technique.
It could be germ-free animals you use.
So when you do the protocol, the rapamycin protocol on these mice, it doesn't work. They're not protected from that second infection.
Right.
So then the next thing, this suggests it's an antibody mediated.
Yeah.
So Dickson, what would be a really quick and dirty experiment to show that?
I would transfer the immunity somehow.
You're awesome, Dickson.
Spot on.
You think I didn't read this paper, don't you?
Yeah, I think you didn't read it.
No, I read parts of it, but I didn't read that part. But that's what I would do next.
Yes, they did that. They took one of these rapamycin-treated mice where they had gotten the response. They took their serum, gave it to another mouse, and it was protected against the H5N1.
Right.
So it's something to do with antibodies.
Son of a gun.
It looks
Now
like- it
all we
looks
have to
like
do
we're
now.
getting broadly protective antibodies against a flu virus.
That's right.
Apparently. And
all we have-
rapamycin is somehow doing that,
Right.
right?
We don't know which class of antibodies they're doing.
Ooh!
Oh,
Should that be now the next question?
he is doing
Before
cue
you do
cards.
that, I just wanted to point out that
Yeah.
this,
No, Vince is a fast writer.
I'm sorry.
Go ahead, Kathy.
you can challenge as much as 6 weeks after the rapamycin and still see the effect.
Right.
So that
Yes.
gets at that long-term question that
Right.
I think
Yes. At last.
Dixon was alluding to also.
Right.
Yeah, as long
Thank
as you treat, around the time you give the antigen, it will last, right?
Yes.
Right.
All right. Then next they turn to the antibodies.
Sure.
They looked to see if there were antibodies in the sera of these mice that
Passively transferred.
could be the original rapamycin-treated immunized mice, and if they could distinguish, rapamycin versus PBS mice versus by hemagglutination inhibition assays or neutralization assays, and it really didn't tell them anything.
Actually, I really like this table. Okay,
you can-
because they take serum
Right.
from the immunized mice and check it for hemagglutination inhibition or microneutralization
Right.
on the immunizing virus, and they're hot. They do hemagglutination inhibition and microneutralization. And with the challenge virus, they don't do that. Okay, even though they're immune to that virus. So that suggests to
they don't
me that
detect
the
it,
antibodies are not reacting with the HA, or at least with the neutralizing epitopes, or
That's a good
the
point.
normally reactive epitopes on the HA.
Yeah.
So when I first looked at that table, I thought, what is going on here? Because these antibodies aren't working. But they aren't working in those specific tests that look at very particular epitopes on the HA.
Right.
That means that what is working, that we know there's antibodies working, but they're reacting with something different,
Yeah.
right?
So there you would have gone home, right, Rich?
Oh, yeah, I published. No, I published that. Or try to publish, and then they come back and say, I want you to do 1,000 more experiments, which...
Right.
So following up on Rich, then they said, what are these antibodies binding to? Because we know they're protective.
Exactly right.
So they did an experiment that made me tired when I thought about it. They took maybe this is not the experiment. They basically bound virus to plates. Okay, this is not the experiment that made me tired. They bound viruses to plastic surfaces and asked
What antibody species sticks to
what's
them.
sticking to them.
Yeah. Pull down this.
In this case, they can they find that in fact the immunizing virus, you can get lots of IgM binding to that, but the IgG is much lower. Mm-hmm. Okay, so a lot of IgM antibodies binding to X31, but not IgG.
Right.
Keep that thought in your head, Dickson.
I will.
And then they said, is there a problem with class switching? Because if you see a lot of IgM and not a lot of IgG, and you would expect by that point that you're getting IgG, they said maybe there's a problem with class switching. So that's the procedure during antibody generation where you go from an IgM to something else, right? Yeah, IgG or
that's right.
One of the others.
IgA.
Right. So it's for the people who might not know that much about antibodies and so forth. The first antibodies that you make are generally this IgM type, and they may not be very strong against the protein. And then there's DNA rearrangements that gives you this class switching from IgM to IgG. And then those may have better affinity.
Right, affinity. Because they undergo also somatic mutation in the germinal centers, and they can get higher affinity. And we talked about this with Gabriel Victora.
Right, I'm going to look up that episode because that would be great background for this whole thing.
We'll put that in.
Concerto in B.
Yes.
Now, the IgM antibodies can multimerize, so that can make up for the low affinity. It's one of the ideas that we think that by joining like 5 IgMs together, you can get a higher avidity, make up for the initial low affinity before you get the somatic mutation and selection and so forth.
Right, because those are generally sort of your rough draft antibodies before you've generated more mature ones that can work better on their own.
Right.
So they measure the different classes of antibody, which you can do, and they find that only IgM increases in the rapamycin-treated mice. In the PBS-treated mice, you get IgM, IgG1 and 2, etc. But in the rapamycin-treated mice, apparently you're restricting it to IgMs. Yet those are clearly given some protection.
TWiV 161, Concerto in B.
That was a great episode.
Required background for this episode.
Yeah.
That was a great episode. I learned a lot in that one.
I learned heaps on that episode.
Yeah, I was accessing that all the time as I was reading this paper.
Let me ask another naive question then. Of course, I'm good at that. On a second challenge in these animals, did you say there was or was not an anamnestic response?
You know, do you want them to measure the antibody response?
Again, I want to know if it comes up.
They did not do that.
It would be very interesting to know if,
It's only 28 days after immunization, right? So it's not enough time for memory, I think.
Well,
so there's still some persisting antibodies from that first immunization. Whether memory does something is an interesting question. You would have to look, months later, I guess, right?
Well, they did do a challenge as Kathy pointed out, that was much later, right? Weeks later.
6 weeks.
And that should be, I think the conclusion from that was, yes, this is a memory response.
But to IgM antibodies?
That I don't know.
Or not?
I don't know.
That was my question.
Yeah.
Because usually an anamnestic response to a second challenge is an IgG response.
Right.
I think Dickson wasn't reviewer number 3 on this paper.
Because that would be do you get... Okay, fine.
So the class profile in that later challenge, that's what you want to see.
Yeah.
That was not done.
I would guess that so it's possible that this IgM bias would be maintained in the memory cell population.
That's really an interesting question.
I don't know.
Because first example of an anamnestic IgM response.
That doesn't make sense because rapamycin has to do the inhibition of, as we'll see right now, it's inhibiting class switching. So if there's no rapamycin around 6 months later, I don't know if you'd get the same bias, right?
Right. It's very interesting because when they did the later challenge, they still got just 28 days of rapamycin, right? Yeah. And then stop that and wait a long time and see if you're still immune to the challenging virus. So there's a very interesting question as what would happen during that period of time in the absence of rapamycin to sort of the immune profile.
Yeah.
Clearly they're still immune to the heterologous challenge virus, but we don't really know anything about the class of the antibodies or anything like that. That's an interesting question. That's the next paper.
Now they do some experiments to see how this is working, and one of the things they look at is the formation of germinal centers and the proliferation of B cells. So germinal centers are areas in lymph nodes where the B cells undergo class switching and maturations and then selection for high affinity antibodies, somatic mutation, and all that. And what they find, they can look at the germinal centers physically in the lymph nodes that are draining the peritoneal cavity.
You could do that.
So let me see if I got this right about germinal centers, because this all goes back to this concerto in B. It's like a B cell that has been now stimulated with an antigen and is making IgM can, in effect, colonize a secondary lymphoid tissue like a lymph node and proliferate like crazy. And so you can stain them with appropriate stains. You get what is a colony of B cells that is a germinal center. And that actually has some differentiation that goes on within it. And then within that, you get, as Vincent said, two things going on, really. The class switching and the somatic mutation, where there's a lot of changes to get a better reactivity are tried, and the ones that don't work are thrown away, and the ones that work better are kept and amplified.
That's right. That's right. The ones that don't have higher affinity are thrown away. They're apoptosis or something like that. Yeah.
So there's a lot of selection for higher and higher specificity antibodies, which basically gives you a better antibody response, but is going to, relevant to this paper, narrow the specificity to something that's very specific for something, rather than starting out with something that's broader, potentially broader.
They prevent with using rapamycin.
Right, rapamycin.
Yeah. So
it keeps you as a generalist.
Yeah, rapamycin inhibits. They found that when you give the mice rapamycin, you inhibit the germinal center formation.
It's very cool.
And the proliferation of B cells is also.
Yeah. So is that also true in the spleen, by the way? Did they look in the spleen also?
They didn't look in the spleen.
Because that's a big germinal center.
They just looked in the mediastinal lymph nodes.
Yeah.
And they have a
mediastinal.
Mediastinal. Well,
that's how I've heard it.
Yeah, I agree.
That's how I've heard it.
Mediastinal, that's right.
I like mediastinal. My father used to say mediastinal.
He was Italian.
He used to take them out all the time.
And my sense is that this aspect of, or I may be wrong here, this aspect of rapamycin effect, is this new in inhibition of germinal center formation?
I think that's a new finding.
Yeah, and inhibition of class switch.
I believe so because they do some really interesting mouse genetic experiments later that are addressing that.
And we don't really know what the basis of that is. We don't know how rapamycin would do that. And we don't know whether you don't form germinal centers because you inhibit class switching, or you don't do class switching because you don't form germinal centers.
No, well, so there's another wrinkle here. So there's this deaminase called AID, right, which is important for somatic hypermutation, but also is required for class switching. And there's some, and they actually provide some evidence later that mTOR is interfering with AID. So if you inhibit mTOR with rapamycin, you're basically inhibiting AID.
Right. Okay.
So that may be the mechanism, and I think that's new. But
yeah, at least the rapamycin reducing formation of germinal centers and inhibiting class switching are both listed in the abstract as if they're new findings, and that's the way I took it from reading the paper.
Okay. Yeah. All right. So that's
also, it seems like if this wasn't a new finding, they would have probably gone right to that and not spent so much time dithering around with all these other things that turned out to be false leads.
They do an experiment to show that rapamycin actually inhibits class switching. It doesn't just inhibit B cell survival, which would be one way to block class switching, but it actively, it doesn't affect B cell survival, but you still get an inhibition of IgG formation.
Yep.
And they do that by, again, measuring in vitro. They use an in vitro assay with cultured lymphocytes. And
yeah, but my only gripe about that, and not that they can do anything different, is that that's an in vitro assay.
And they use
whether all their other
Yeah.
measurements are
In vivo.
in animals. Okay, so you're saying that what we're observing in vitro is the same thing that would go on in the animals. Fine, but there is a shall we say caveat there.
Yeah. And the experiment they do is to use lipopolysaccharide treating B cells in culture, and that caused class switching to occur, right, from IgM to IgG, and they show that's inhibited by rapamycin. So it's even a different antigen on top of it. But,
Right.
but I'm sure that's the next, series
Sure.
of papers. So then they did a series of experiments, which I don't think we really need to get into, which are absolutely cool, but involve deleting one of the proteins that make up the mTOR complex and looking at the effect of rapamycin. And the conclusion is that this one of the subunits of mTOR is absolutely needed for transcription of the gene encoding AID, one of the isoforms of AID. So the idea is that somehow rapamycin is interfering with AID, and that may be how class switching is prevented.
Right.
All right, the next experiment is the one that made me tired.
Yes.
So they took the sequence of the HA protein and they made overlapping peptides, 20 amino acid long, overlapping 15 amino acids.
Wow.
I guess you could. It's not really phasing, but you could. And they did the whole protein, all right, and then they stuck these peptides on a surface, and then asked which ones are bound by the antibodies in these mice, in these rapamycin.
Here.
Right, you got
That's
it.
a great experiment.
They want to know which
Yeah.
are the epitopes,
Sure,
right?
You bet.
Of course, it's great, but it's only looking at HA,
That's true.
and it could be other things involved.
Good.
But
Right.
then you say, where are the antibodies binding? And it is a nice picture that shows this beautifully because they color the epitopes. The rapamycin, induced epitopes are red, and the PBS induced epitopes are blue, and they're totally different.
Yeah.
And rapamycin gives you this totally different epitope representation in the antibodies in these mice.
Very interesting.
It's really cool. And they even map these on the HA structure because you know where the structure is solved and you know where each of these peptides are. And the red ones are, some of them are on the head, but many of them are on the stem, the conserved stem of the HA molecule.
The conserved stem.
So the HA is a stalk-like
no.
molecule, right? It's got a stem and a globular head.
Yep.
And most of the antibody response, the virus-specific, say, antibody response occurs to epitopes on the globular head, but the stem has the broadly conserved sequences. So a lot of these are down there.
And the head varies with each strain.
Head is highly variable, yeah.
Okay, so that goes back to my other question, then, which was, are we dealing with a common antigen between all these strains of influenza?
So to a certain extent, this stem has some of those common antigens in it, yes.
Right.
My guess is if you looked at other influenza proteins, you might find reactivity to some common antigens there, or sort of changed reactivity.
So
Right.
do you account for... I'm sorry.
This goes back to the table where it was found that the rapamycin treatment gave an immune response. It gave an immune response that was protective against the heterologous virus, but apparently the antibodies that were made did not react to the normally reactive parts of the hemagglutinin, because it wouldn't. The antibodies would not inhibit a hemagglutinin. Were not reactive in a hemagglutination inhibition test or in a microneutralization assay against that heterologous virus. So this profile of altered specificity of the antibodies fits with that phenomenon that was seen in the table. Cool.
So these antibodies that neutralize the H5 after immunization with H3, these are on the surface and they're on the stem.
Mm-hmm.
All right. So and this is different from. So these are the rapamycin specific or rapamycin induced antibodies, which are different from the PBS induced antibodies. They're all on the exterior portion and on the stem.
So we know the specificity of the antibodies in this protocol is changed. I don't know that, well, we don't know that it's those antibodies that you're looking at in this experiment that are protecting
Right. We don't.
against the infection. We don't really know what the what's giving protection aside from the fact that it's B cells, antibody mediated.
So there's another question here too, and that is that the rapamycin induced immune responses are all IgM,
Yeah.
and the PBS immune responses are not.
No, they go to IgG.
Correct.
So
that's
they class switch.
The point is that what about the IgM antibodies induced early on in the PBS mice?
Yeah, well,
What do they look like?
Yeah,
Yeah, they didn't look at that.
I want to
But
know.
you would guess that there would be some that would overlap with these, right?
We hope so.
Yeah, because what rapamycin is doing essentially is freezing
Sure,
them
exactly
at the IgM.
right, exactly right. And that's
but
another
you just-
question. What about the timing of this, so that you could, vary the time that the animal was exposed to rapamycin to see if there were some graded responses, and maybe you could get a gradual switch to IgG, or maybe a little bit of switch. The closer you got to the immunization after giving rapamycin, so that you could start to look at what the mechanism was for the freeze.
I think in their figure they do show some of the early PBS reactivity being to similar peptides as I see with the rapamycin for IgM.
There you go.
So,
That would have to be an early IgM response in those groups too. Yeah.
right.
But
Right.
those don't get selected out for affinity maturation.
Yeah,
Right.
that doesn't happen with the
Same
rat.
rate.
So-
So, yeah.
This almost looks like a viral escape mechanism.
So the affinity maturation doesn't occur. So
Right.
you only get the low affinity IgMs, which tend to be the ones that are cross-reactive.
Right.
And the high affinity ones, which happen later and you get
Oh, very
IgG,
interesting.
are generally directed against less conserved epitopes on the globular head, for example.
Exactly.
Yeah, so
Those
you
are-
can have broadly neutralizing or high affinity.
But you can't have both.
Pick one.
That's right.
Yeah.
Right. Right.
This comes back to
Love
I guess it was Dickson's question earlier on, and that is, what's the profile of antibodies if you now look a lot later? Have they matured to IgG,
right,
and what's their specificity, et cetera?
Right.
So why is it that the high affinity maturation ignores the
Exactly.
the cross-reactive epitopes,
that's
right?
You'd
Why?
have to. Well,
I know you can't ask why questions, but
You can't.
what is the mechanism of that?
what are the binding
Yeah.
sites of the high affinity antibodies, and what amino acids are involved?
Well,
Maybe
as I
in-
recall, the maturation has to do with, among other things, antigen persisting on dendritic cells, follicular dendritic cells,
Yeah.
and being there to be sampled all the time. So it may have to do with exactly what parts of the antigen persist in that fashion for during the maturation process. Pretty good, huh? Did I get that?
I love
good.
it. I love it.
Could be.
I hope Gabriel's listening, or somebody. We're going to get some mail on this, I hope.
Well, that's okay. We can all learn.
Oh yeah, to
But,
fix this up.
The
We'll
general idea is correct. Rapamycin is altering. It's preventing class switching, so
Right.
you get mainly IgM, and it's altering the antibodies that are recognizing certain epitopes.
Exactly.
So the real question is, and we don't know how these neutralize. That's the other thing which Rich was pointing out before. We don't know how these work, which would be really interesting to figure out.
Right.
So you can imagine now taking some of these and trying to sort that out. How would you use this? Assuming that, how do you use it to help our vaccine making?
You're not going to use it on me.
Well, I don't think the idea is to give people rapamycin, but
No.
to learn from these epitopes,
Yep, sure.
right?
Exactly. What if you could clip off the head of the HA, leaving the rest of the viral protein intact, and
Yes.
using that as a vaccine?
Yeah, so Peter Palese has done that.
Really?
And you get it cross-neutralizing antibodies, but they're low levels, they're weak, okay?
Yeah.
They're not great.
Got it.
And so another approach he's taken is to take the stem and put a totally different head
Sure.
that people have never seen before.
Okay.
Okay, so that you don't get a memory response against the
Got
head,
it.
because
That's
that's the problem. When
Sure,
you get
sure,
memory
sure.
against the head,
Right.
then you swamp out the stem.
Exactly.
So now you get a primary response against the stem, and then you get good So that's why I say that there is some progress in these cross neutralizing approaches. But now we have new epitopes from this kind of
study. Or just
study.
a synthetic antigen. I mean, you could just come up with that instead of the virus itself.
Maybe.
We have new epitopes from this study, and we also have a better view of what's probably going on in the immune system as this response evolves.
Yeah.
Exactly.
And where it's going wrong.
And the interesting thing is that HA inhibition or microneutralization is not measuring necessarily the antibodies that you want.
Right.
What's
we
the matter?
lost Richard, I think.
Did we lost Rich?
Did you lose me?
No, you lost your picture.
Just the
I get panicky when I see your picture disappear.
the
Richard.
monitor just went to sleep, Dickson. It's okay.
It did, okay.
So is that correct? What did I just say now? I forgot.
Oh, I'm sorry. That was my fault.
So the microneutralization and the HA inhibition do not measure the right antibodies for this. In this case, right? Because they didn't see a difference in the neutralization or HI ability of PBS versus rapamycin treated. Yet those protective antibodies were there, right?
Yes.
So there's something else. There's some other mechanism that's not being measured by those two assays.
Right. Those assays are measuring what you usually get. They are not measuring what you actually want to get.
Yeah, they're getting antibodies against the globular head that definitely inhibit plaque formation and hemagglutination.
One
Right.
possible outcome of this that might be very useful is to develop monoclonal antibodies against those epitopes in the stem to treat patients suffering from H5N1.
You know, the problem with that, Dickson, is that it costs $25,000 to treat them. It's really expensive. Okay?
If you're using money as your argument, then I would say I don't.
But who's going to pay for it?
No, I don't know how expensive
Right now,
that is.
a company makes monoclonals to treat babies who get respiratory syncytial virus infection. Okay, this is highly lethal, first 6 months of life. The course of treatment is $25,000 because the monoclonals are humanized. They're very expensive to make. So it's a good idea, but I don't think this is therapeutically useful approach. It's too expensive.
And I don't know if that would necessarily work.
Well, you need an animal model.
Yeah.
The problem is that we don't have an animal model.
For influenza?
Yeah.
Well, you have to test it in what you have, and then you have to do a clinical trial. That's the only way.
Exactly.
That's the only way. Anyway, it's a cool paper, isn't it?
Very.
Thank you,
It is.
Kathy.
Yeah.
Very nice.
Thank you very much.
Before we leave,
Yep. Are you gonna,
I just want to point out that they do plaque assays.
Oh yeah.
I don't actually see where the results of a plaque assay show up in the figures, but they got it in the methods. And the other thing, this is sort of a triviality, but I was because I'm always interested in the methods, in particular when they're good old classic methods. The doses of viruses that they use in these experiments are the flu are doses that are the units are EID 50 or egg infectious dose 50.
Yeah. Right.
That strikes me. My understanding of the way you do that is you do a serial dilution and infect a bunch of eggs and then sample the eggs and see if they've got hemagglutinin.
Right.
Okay? That strikes me as like more laborious than a plaque assay or an HI hemagglutination inhibition or something. Why use that test? Do you have any experience with this?
In my flu career, I never used an EID 50.
You never did an EID
plaque
50.
assay or HI or HA, yeah. I don't know why you would do that. A plaque assay would be much, and maybe in this, I don't know, maybe not at all cases the viruses are plaquing. I don't know.
Okay. All right.
Don't know.
Just curious.
All right. Let's move on to philosophy, Dickson.
Great.
This is actually stimulated by a letter from our friend Chris Sullivan, who wrote this week, Howdy TWiV hosts. Better late than never, a well-written one-page article from The Economist magazine on the problems of, quote, crap, unquote, in our biomedical publications. I don't believe in all of their proposed solutions, but a topic that's worth a good thinking from all of us. And he sends a link to an article in The Economist. Scientific research has changed the world. Now it needs to change itself. How science goes wrong. Many thanks to all of you. Still enjoying a TWiV bump from being a guest on last week's show. So this was his delayed pick of the week.
Okay.
Right? He didn't have one
Right.
during the show, so he sent it, and this happens to be an article that a few other people have sent and has been generating some interesting discussions. So I thought we would talk about it. There are actually two articles in this issue of The Economist, October 19th, 2013. One is called, as I said, Problems with Scientific Research, How Science Goes Wrong. And the other one is Trouble at the Lab. Scientists like to think of science as self-correcting. To an alarming degree, it is not. So the claims here are things that we actually talked about during our discussion of those papers on reforming science a while ago. That a lot of science is wrong, and the reasons are complicated, but have to do with things like the dwindling amount of money, competition, publication, rushing, promotion, getting jobs, etc. And really the theme in these two articles is that a lot of science is wrong, and that we need to be better with statistics. We need to change the way papers are reviewed. There's too much pressure on having really hot results and so forth. And these struck me. I mean, these kind of articles bug me in a way, because I look at the paper that we just did, for example. I mean, most of the stuff I look at in my, and I have looked at in my career, papers in virology and cell biology. I wouldn't say that most of them are wrong.
Right, I agree.
I don't know if it's our field, but like the one we just did, everything is beautifully controlled, right? All the controls are there. And, aside from overt fabrication of everything, which happens but really rarely, it's not wrong. And I think that's been the case. I know that Condit's never published a wrong paper, for example.
Never.
So I don't know how to interpret this, because a lot of the papers science is not just virology and molecular biology, of course. It is other things like psychology and other kind of softish sciences where it's really hard to draw conclusions. You depend on a lot of modeling and so forth. So I would like to hear what everyone thinks about this.
Well, I have a similar reaction. First of all, I would say that 35 years ago, my thesis was based on a Nature publication that turned out to be very flawed. And I made the mistake of not looking at that publication closely enough, or trying to reproduce the major findings in that publication before I pressed forward with my own research. And one of the major lessons that I taught myself in that process is that, not everything that's published is correct. And if you are going to take some published piece of work and as the foundation for what you're going to do, it's very important that the first thing you do is to try and reproduce the major findings in that work with your own hands, so you're sure it's right before you move forward. Yeah, that's right. So the point there is that that's nothing new. And it's part of the whole process. I think that one of the things that's happening, it may be worse now. It's not something that, when I look at the literature, for example, I look at this mTOR diagram that I pulled out, this pathway with a billion blobs in it and a bunch of arrows going everywhere with all of the stuff that happens in all these pathways. There's hundreds of these diagrams around, and I wouldn't be at all surprised if a lot of the background literature that generated these extraordinarily complex diagrams was flawed. I won't say wrong, but flawed, because they're based on small assumptions changes and everybody, pursuing his little pathway with his little inducer and his little outcome, okay, and it just all kind of gets messed up, okay? So there's a lot of stuff that needs to be sorted out, but I don't see it as a huge problem, okay? I think in the long run, the stuff that's true floats to the surface, gets verified, gets sorted out. I don't think it's that big a deal. I think where it really becomes a big deal is with people in the commercial sector who want to do big clinical trials on a drug, and the preclinical trial data is flawed. And that's where you see this come up in these articles where they say, we looked at a bunch of preclinical trials and we looked at 60 of them and only 9 of them were true. That's a problem. Okay, so in my experience, in my own personal experience, I don't see it as a huge problem. I think it's probably a bigger problem than it used to be. And the conclusion that we came to before, by the way, that was in TWiV 184, Reforming Science, was that the biggest problem was not enough money, therefore more competition, and so people are pressured to publish stuff without rigorously following through and making sure it's right.
Or small snippets of a larger story, and they dribble it out.
So 2 comments I have. One is that this Economist article points out several different cases where papers were sent out that intentionally had flaws in them, and the reviewers were instructed that they might find flaws, and they didn't do so. But they say, as well as that kind of thing, reviewers can't be expected to spot deliberate falsifications if they're carried out rather subtly. And to that, Peter Palese wrote a foreword for Sendai virus that he sent me, and he describes how there were papers early on for the infectious clones for measles virus, and he was the reviewer. And he said, I, as a careful reviewer, could not see any problems with the manuscript. If someone wants to mislead you, it can happen easily. Mm-hmm. So while we can feel like we have a pretty good handle on things that we understand, we have to rely a lot on reviewers who are knowledgeable about these things that we're not so knowledgeable about. So, for example, the experiment in the paper that we just described that Vincent said made him tired, I was overwhelmed by reading the materials and methods for how they did this analysis of these peptides. And by the end of it, I thought, well, I just have to take their word for it and hope they got the right reviewers, because it's got a lot of statistics, it's got a lot of software and stuff in terms of how they came up with these dendrograms. And so we aren't all experts on all of these things.
That is a great point. A paper like that, I would be really surprised if it got a completely thorough review by all the reviewers. It's just too much.
Right.
Which almost says that maybe we shouldn't peer review papers. We should just do what other fields do, is just publish them and let them get all sorted out.
Well, this is part. I think we should peer review papers, but it's part of where I'm coming from on this. I guess I kind of assume that there's going to be, based on my own experience, going back 35 years, that there's going to be a certain amount of stuff, maybe a large amount of stuff, that's not right, and it's part of the deal. Over time, the stuff that's really correct will surface, and the rest of the stuff will disappear. And
what's important?
It's too bad that there is a lot of stuff that's not right that's published, but
I think that's the nature of science. I'm not sure.
The nature of science.
I'm not sure you can, aside from fraud and misleading. I think it's the nature of science that a lot of it's going to be wrong.
Say Freud or fraud?
I don't think it's bad.
Did you say Freud or fraud?
All right.
And it's just, if you're going to invest a lot in a particular protocol or experiment or something like that, you have to understand that it could be that some of the foundational material is going to be wrong, and you have to take the time to make sure that what you're going to do, that you try and reproduce that, and what you do is based on stuff that's correct.
Right.
But the degree of wrongness varies radically from field to field.
Right. Right.
Yes.
I agree. Yes.
And it is, I think, going back to Vincent's original point, just looking at this paper we just talked about, there are some degrees that could be wrong, And Kathy made an excellent point with the statistical analysis on that antigen map. My eyes totally glazed over. I said, okay, there's the pretty picture. And I'm going to go with what they're showing in the picture because I am not qualified to evaluate how this whole darn thing was done. And yeah, I'm sure that the reviewers were not all qualified to evaluate all aspects of those experiments. They couldn't have been. But that's one piece of this body of work that we've just discussed where you look at many, many, many of the other pieces of it, and they simply cannot be very far wrong.
Yeah.
Because so much else about the world would have to be wrong for those experiments to have yielded those results. And put another way, if you've got an effect that's 5 orders of magnitude, you don't need a statistician to tell you the experiment worked.
Yeah.
Right.
So the way this gets covered, unfortunately, in a place like The Economist or any other general interest magazine is most of science is wrong.
Yeah, right.
Well, no.
We already knew that.
Much of some fields of science has a greater range of error than we'd really like to see.
Right.
And we are probably making decisions based on results that are not as reliable as they should be for the magnitude of those decisions. That is especially true in things like pharma, where in the case that Rich is talking about with doing an experiment and kind of investing some of a graduate student's time in presuming that this previous result was right, and then your experiment's going to fail, and you're going to wonder why, and you're going to troubleshoot it, and you're going to figure it out. But you're in a lab where you can fairly easily replicate earlier work done in another lab, and if you can't, then you can probably troubleshoot it. You're not running a 50-center, 3,000-patient phase 3 clinical trial that nobody's ever going to be able to repeat. So I think a lot of the problem stems from these very big decisions that are being made on data that would be very, very hard to reproduce.
I think that's why this fellow John Ioannidis, he published this paper in 2005. He said most published research findings are probably false because the ones that depend on statistical analyses and power and so forth, a lot of those it may be true, but a paper like the one we did does not depend entirely on statistical analysis, but a step-by-step interpretation of pathways and understanding what's going on. Yeah. Right? So I cannot accept the conclusion that most published research is wrong. And his approach is a statistical one based on models, which I would say don't really measure what's going on in a lot of the fields of science.
Yeah.
Right. And you think about, for a lot of things, we talked about this a while back, that it used to be that we were mostly doing molecular virology and you're looking for a band on the gel or not a band on a gel, or the sequence is ATCG and you can read that off the gel. But when you're doing things like pathogenesis and you're starting to work with small number of mice or animals, whatever, that you're doing these necessary statistical analyses, which have some false positive and false negative rate that you have to be aware of.
Right.
So it's a little bit of a difference in the shift in the kinds of science. And I agree with you that the kinds of science that we're mostly looking at are not relying on a lot of these statistical kind of things.
Statistics is becoming more and more a factor in the stuff, in the kind of stuff we do, I think. And I think it certainly is true that that's subject to problems, and there are too few of us who know how to either evaluate or use the statistics. So one of the recommendations that's very commonly made is better education in statistics, and I certainly agree with that. I think that needs to be done. I'm completely ignorant when it comes to statistics because I was raised with there is a band or there isn't, or there's 2 orders of magnitude change.
Yeah, the kinds of experiments, as we've said, in pathogenesis now require statistics more and more, and that's fine. But I just don't agree with this sub-headline, science is self-correcting to an alarming degree. It is not. I just don't agree with that. I think what is important will be looked at by others, and they will attempt to reproduce it. So the paper we did today, even if there's something wrong with the peptide antibody studies,
that'll come out.
if this is an important result, other people will look at it. If it's not true, that's the end of it. But that's how science works.
But in a field like that, where it's laboratory research, looking at it means they're gonna try to extend those results.
Right.
And if you're in any hot field of laboratory research, someone else is going to try to extend your results, either to compete with you or to do something different. And when they do that, just as a matter of routine, they're going to reproduce what you did, or they're going to do things that rely on your findings being true.
Right.
And if their stuff doesn't work, they're going to start picking apart exactly why. So they're not going to be able to publish negative results, but everybody has to replicate what came before in order to do laboratory science of the type that we most often talk about. The problem arises when you're doing stuff that can't easily just be rerun. You know, the next person who does a drug approval trial on something isn't going to repeat the previous one or do something that relies so heavily on its findings that things simply wouldn't work at all if that were wrong. So that's not a routine part of that type of science.
Dickson, you have any thoughts?
I sure do. And then, I mean, I've lived through an era of what is the genetic material versus I now know what the genetic material is. And you could fill a room with the books and journal articles on what is the genetic material. And there's a single sentence that appears at the end of an article in Nature that says it's probably DNA. And then now there's another giant room filled with how does DNA work, and then you'll boil that down to a few paragraphs at the end also. So that's one issue. The other issue is that, you know, we've got our heads buried in our petri dishes and looking for plaques. And if you look out the window, what do we start every broadcast with? We talk about the weather and what the humidity is and everything. And so the International Climate Change Panel that is obviously very, very much concerned about, is the climate changing faster than it should? And are we the cause of that rapid change? Those are experiments that are not possible to do, so you have to use statistics in order to make your points. And that's where these, popular press journals pick up on the fact that not everybody agrees on this, not everybody agrees on that, so therefore we can't trust it because it's unreliable, because we rely on statistics and observation rather than on experimentation. But it's still valid if you do the proper observations and you use the right statistical analyses. Again, I'm with Richard on that one, but I trust the reputation of people that are cautious. And that's the point. If a person is not cautious and always says something that's sensational in order to raise the bar for their reputation, I'm worried. And if they cautiously say, you know, we might have found something out here that has some validity because we've observed this 15 times in 23 different places, and we're darn sure of it in the sense that we know what's going on, we just don't know how much of it's going on. I trust those people much more than I trust someone who says, I have a 190 base pair fragment from the feces of a flying mammal that says that. All right, I won't go any further, but the point is that you shouldn't say those things like that. You should do the experiments that allow the other people to do them. Why don't you do them first so that you can say something better and more secure about the results that you've gotten? So two things. One is observational science, where you're bound and determined to use statistics, and the second is experimental science, when you can actually tweak something and watch it happen. And when you tweak it, you should only try to tweak one thing at a time so you get a yes or no answer. You get a band or you don't get a band. You do. Go ahead, Alan.
I don't trust people generally.
Well, you remember my statement is trust science, don't trust scientists.
I get where you're coming from. But in a case like purely observational research, what I look for is, are the people in that field making testable predictions and then testing them? And testable hypotheses. And in fact, what you find in something like climate science is probably something that that pharmaceutical research and biomedical research could learn a lot from. Maybe. Which is, you know, hey, if the planet was not getting warmer, we would expect to find evidence that it's actually more steady in geological features that are more steady, like glaciers.
That's right.
So let's go drill some ice cores. And then they go and drill some ice cores.
They do.
And they say, oh, darn it, that's also saying the same thing that we were trying to. All right, well, we would find this evidence in the fossil record by this. And so let's go look at some and you just keep repeating this sort of thing over and over, and you keep coming back to the same answer. And eventually you say, folks, this is what's going on. But we don't do that in statistically based biomedical research, and we don't do that in psychological research or so-called soft science research. What you get in those cases is somebody comes up with a result, and then other people just kind of assume that it's true.
Right. And I remember the issues surrounding the anti-angiogenic factors that are created by the formation of blood vessels. And there was a very famous Harvard angiogenicist whose name I'm completely blocking on right now, but they published a book in his honor even. And no one else could repeat his results. And so his approach to that was, okay, come into my lab, sit down at my bench, and do my experiments, and then you tell me what you get. Because, I'm not lying to you. I want you to see what I got. And virtually everybody who elected to do that found the same results that this guy found. So at that point, it was something wrong with the other labs, not his.
Is that Judith Folkman?
It is Judith Folkman. Exactly right. Thank you. So, he was an honest man that got an honest result that no one else could replicate until they went into his laboratory. And then they went back and said, oh, I see what's wrong. Here it's the kind of Teflon that I'm using from my distillery to make my distilled water from, or something stupid and simple like that was a basic difference that no one ever recognized as important until that point. So could be things like that. This
John Ioannidis had a quote in one of his articles, which I think is worth using to wrap this up. He said, scientific investigation is the noblest pursuit. I think we can improve the respect of the public for researchers by showing how difficult success is. Confidence in the research enterprise is probably undermined primarily when we claim that discoveries are more certain than they really are, and then the public, scientists, and patients suffer the painful refutations.
Yeah, that's a good quote.
Yes.
So that's good, but I don't think that publishing a paper saying most results are wrong helps. And these two Economist articles, by saying science is not self-correcting, I think that's an incorrect conclusion, because as we have said, it depends what science you're looking at.
Right.
And in the end, I do think that what we do here in our little circle is self-correcting, and what matters will float to the top. And
absolutely.
You'll know if it's right or wrong.
The last two paragraphs of the longer Economist article has some quotes or near quotes from Bruce Albert's testimony to Congress about, the system's not perfect, right? So there could be changes that could improve it. And I thought that Albert's comments about the things that could be done are really quite good. So if you look at these articles and do nothing else, look at the last two paragraphs where Alberts gives his recommendations. I think it's very good.
Yeah, except he says journals must do more to enforce standards, and I think that's not a good way to go, because they are not a disinterested third party.
Yeah, but then your comment about everything should get published doesn't fly in that.
Why not?
Well, because then how do you select out the stuff
Well,
that's
the
good?
stuff that's important will get replicated. The ones that don't matter, they'll just be out there. And, the one of the problems is tenure committees won't know how good it is. Well, you know what? You can read the darn stuff instead of just seeing that it's in Science, Cell, or Nature.
No, that's, that doesn't really work on tenure committees.
No, Dickson, the tenure committee does not do its work because it says how many cell papers.
They can't.
Wait a minute. How many cell papers does this person have? What you need to do is read the papers and decide whether they are good or not. And most tenure committees are not qualified to do that. They're too broad, and it's a lot of work.
Exactly.
But those are not good excuses for having a third party do the work for you. I think
Kathy had a comment as well.
Yes, Kathy, sorry.
Just to the point of, we'll just publish everything. But there's going to be some garbage that waste their time commenting on. And that's just going to stay there. For the public to jump on and post on their website.
You mean like vaccines cause autism and stuff?
Well, I don't say you just put it anywhere, but like PLOS One will publish stuff that is methodologically sound. In fact, it's mentioned in one of these articles, right? Well, and in fact, they say they have a 50% reject rate because a lot of what they get. And I think, so you go there, you can see there's a lot of stuff that's not interesting, but there's a lot of compelling, interesting stuff. People will pick up on it if it's right. Gets validated if not, and then you have a tenure committee saying, look, this person published this seminal paper in PLOS One and we think they should be tenured, and I think that would work fine. I mean, you can't change it overnight, I understand that, but I just don't think having the journals be the arbiters works either, so there's got to be a better way. All right.
Dead silence followed.
I'm sorry that
No, we're just talked out.
The weather outside today is a little...
I think we're
going to hear a number now from Count Basie and his orchestra.
I think we should do just 2 emails, okay?
Okay.
Because
that sounds good.
Got a lot. And Kathy, can you take the first one from William, please?
Sure. William writes, Dear Doctors, last week there was a question regarding vaccine trials and whether researchers could challenge their study subjects. In other words, can researchers emulate Jenner and intentionally infect someone with a live pathogen to test their vaccine? The short answer is yes, depending on the pathogen. The most recent and high-profile example comes from a study that appeared in last month's issue of Science. Researchers in Maryland published the results of a Phase 1 clinical trial in which they tested a promising new malaria vaccine. Their subjects were vaccinated and later challenged by being bitten by infected mosquitoes. It was carefully controlled and monitored. Subjects who received several doses of the vaccine were fully protected. In other words, they didn't develop parasitemia. Amazing! From an immunologist's point of view, the correlates of protection are still a little hazy, but if it works, it works. Stay tuned for Phase 2 and 3 clinical trials. And he gives the link to that paper in Science.
Yep.
I also wanted to say that I adored the show and I have so much respect for the whole team. I know how much time goes into it each week, so keep up the great work. Besides, who else will keep me company during those endless plaque essays? Oh my...
Yeah, we talked about this, Dickson, on TWiV, right?
We did.
I forgot that, yes, you could challenge people with with malaria.
You have to have a cure for the disease in case they develop it, though.
Well, it can't kill you either, right?
Well, apparently not.
All right. Alan.
Marianne writes, Hi, TWiV team. Just a couple of follow-ups from your last show. On vaccination, there were a couple of interesting stories this week on vaccination rates and pertussis outbreaks in particular. There's a study published in Pediatrics where they linked the severity of pertussis outbreaks in California to areas with the highest areas of non-medical exemptions to vaccines. And she provides a link. On a related front, the Canadian Centre for Vaccinology in Halifax conducted studies looking at boosting pregnant moms for pertussis so they have high antibody levels to pass on to infants during that critical first 2 months prior to their vaccination. So far, the data look very promising.
Both of those are really cool. This correlation with areas of non-medical exemptions and pertussis outbreaks, I hope, wakes a couple of people up. I doubt it, but, what the heck.
It's a horrible disease to get.
Yeah.
Although Kathy pulled out a rather shocking line.
Well, it says that Wakefield now practices medicine in Austin, Texas.
I didn't know he was in Austin, Rich.
Yes, he is.
We should have gone to visit him.
Yeah, well, I'll be back there. I'll go drop in on him.
Do a TWiV with him.
Sure.
Yeah.
Go ahead, Alan.
2, human challenge studies. Although the types of studies that Jenner did with smallpox are no longer conducted, with good reason, there are considerations with viruses of very different severity. In diseases where natural infection is unlikely to happen except in isolated, often severe outbreaks, I'm thinking Ebola, the correlates of immunity are incredibly important and often have to be correlated with animal studies because conducting a clinical trial based upon epidemiology is unlikely to happen. However, in diseases that are typically relatively mild in normal human adults but can be severe in other groups, the immunosuppressed, young children, and the elderly, challenge studies are possible. I'm thinking influenza, norovirus, and respiratory syncytial virus. These are all conducted under strict controlled conditions and strict ethical approval. They also often provide important data. Both of these fall outside the typical clinical trial progression for vaccines. Keep up the great work. I enjoy listening every week.
All right, so there you go. 2 emails on this challenge stuff, which we had kind of not gotten right. So straighten this out.
And We should have known better. Probably didn't we talk about a place that does?
Over in the UK, there's
challenge,
one.
Yeah,
there's a company who actually
they're a contract company that does these.
Yeah, they wrote to us and said if you're ever over here, you should visit. So
go to flu camp.
Right. All right, let's do some picks of the week. Alan, what do you have for us?
Let me scroll on down here and see what I have. Oh, yes. So this is a... you need to do this on a desktop computer, but it's a virtual tour of the National Museum of Natural History at the Smithsonian.
Oh, it's 3D. How cool.
You go in and you've got a picture of the elephant in the entrance rotunda, and then you've got some arrows down at the bottom, and you can walk around.
So cool.
So you push the arrows and you go walking around the museum.
It's nice.
I've never. It's very cool. I've never been to this museum. I need to do this.
I thought there was a Brontosaurus in the rotunda.
There used to be.
Apparently not.
Now they have this elephant.
Apparently they have an elephant.
It's very cool. I like that. It's working well on my laptop here.
Yeah.
Isn't it cool? Nice.
It's really cool.
Very nice.
Yeah, and you can go with the mouse too. You just click your mouse on the screen.
So we don't have to go anymore.
And you mouse over and it tells you what the arrow is pointing to.
Yep.
It's really cool.
Can you go in the bathroom?
Go to the Hall of Biodiversity.
You can zoom out and zoom in.
You can go to the cafe. I did do that.
Go to the Hall of Biodiversity.
It's virtual food.
You will love it.
Yes,
And
virtual food.
the virtual food, that's right.
Nice.
You would love it.
That's cool. Thank you, Alan. Rich Condit, what do you have?
I have something that my son sent me. It's Mauna Kea Heavens time lapse. So this is a time lapse video of the observatory atop Mauna Kea on the Big Island in Hawaii. It's quite a cool observatory. Photographed by this guy.
Wait a minute.
Dickson.
How do I turn it down? All right, it's off.
Sorry. Photographed by this guy, Sean Goebel, Goebel. He apparently was, he's an astrophysicist who's doing his graduate degree there in Hawaii, and he's also, I would say, even more than an amateur photographer. If you look at his photography website all by itself, it's very cool. But as one of his photography projects, he took this time lapse of over, I guess it's a total of composite about a week of activity on top of the on the observatory, and it's cool. You see all the stuff going on, the domes rotating, and the doors opening and closing. But what's particularly cool is that in order to adjust and focus the scopes, they have to focus on areas in the sky that they identify by shooting lasers up, in particular, that intersect by a couple of different domes, and they can focus on those and adjust all the mirrors and that kind of stuff. So as part of this video, you've got these domes shooting these incredible lasers out into space. So it's just cool looking.
It is very neat.
It is.
Nice. My pick is an event. It is a press release from the Federal Aviation Administration announcing something I've been waiting for a long time. FAA is going to allow airlines to expand the use of personal electronics.
Right.
No longer will I have to stop reading for the first, 15,000 feet?
Yes.
10,000 was their previous
10,000. You'll be able to, look at your tablets and Kindles and all that stuff.
Do you know when that was established, by the way?
I don't know. No.
It was back in the 1950s.
Really?
Yeah.
To restrict the use of what? What did we have in the 50s?
Exactly right. Who knew? We didn't even have any electronic airplanes then.
Huh. Well, you still can't do it on the roll, takeoff roll and landing rolls, but, I think it's cool. I've always had to bring in some paper book or something to read.
You still can't
Although
talk
I did
on your
find,
cell phones either.
You can't talk on the cell phone. That's good. I did find a few times that just sitting there and thinking was actually very useful.
for 3 hours in Washington, D.C., waiting to go home. I know exactly what you mean.
No, and then until 10,000 feet, there is something to be said about a little contemplation.
That's
but right. I welcome this. I think it's cool.
Yep.
Just don't think too deeply about shooting up into the air at 500 miles an hour in a pressurized aluminum tube.
Correct.
Yes, that's something you need to
That's right.
Keep
Just
out.
kind of suspend.
I totally agree with that. Dixon, what do you have for us?
Well, I have something that I'm quite passionate about, as you know. We've had numbers of discussions about automobiles and whether they're fraudulent cars or not.
Automobiles in general are not fraudulent.
So electric cars. So this involves Tesla Motors. I would like you to go to the Tesla Motors website and see the latest developments in their S sedans. And it's a replaceable battery, basically. It's you drive into a recharging station, and it's a hypercharging station. It's all solar panel controlled, and you exchange batteries, and it takes 90 seconds. And remember, I told you last time or the time before that all you have to do is just exchange batteries. So what's wrong with that? Oh, now you need 2 batteries. No, you don't need that many, but you need some, and it takes 90 seconds to make the change, and it takes you longer than that to fill up your gas tank with gas. And the other thing is it doesn't pollute anyway. And then you say, well, where can you go on that? And if you look at their website, they have a map of all the refueling stations, all the charging stations. There's an entire chain. They just completed it from Washington down to Baja almost. And so they're going to do that for the entire country, and I think this would be a fantastic new development. And if you buy their cars, guess what? All of the electric charge refills are free. So you get free fuel for as long as you own your car. That's pretty cool.
Yeah.
That's very cool.
It does produce pollution.
Everything pollutes. Everything
but
pollutes.
not
anywhere near.
Not at the location of the car. It's the coal-fired power plant up the road that's providing the electricity.
Well, no, Alan. They're using solar panels for the
these are all
recharging
solar
stations.
panels.
They're all solar panels for the recharging
they are
stations.
not doing a 30-minute charge of the car with solar panels.
Well,
They exchange the battery.
they exchange the
Yes.
battery. The battery sits there for a longer time, but you drive into one of these things and it's already recharged a whole
I
bunch
think
of
the
batteries.
solar panels are decorative. I'm pretty sure that
No, go.
240-volt main line
You know
going
what?
to this is
We're
not
going to
coming up.
research this. My next pick of the week will be I take that back, as I said last week.
Well, Dixon, the picture they have on the website
Yeah.
is about 100 square feet of solar panels. I don't think that's going to do anything.
That's not going to
No, it won't recharge the battery that you're exchanging. It will.
I understand what the model
Over
is. We're just
time,
a little
it-
skeptical, that's all.
I mean, how many cars are there on the road now that use that technology? Probably not too many.
I'll stick to my 6-speed manual transmission.
We can get you a vroom vroom motor in front.
You know what?
and-
If you want to put a recording of an engine sound,
Yeah, exactly.
that's fine.
Exactly right.
Kathy, what do you have for us?
I picked a FASEB contest that expires, or it ends November 30th. There's a $5,000 prize for someone to make a 1 to 4 minute video to educate Americans about how science is funded. And so anybody who's feeling creative, who wants to help publicize American science, there are some criteria on the web page. There's last year's winning video, which in itself is worth watching. There's an informational video. So those of you out there feeling creative and want to do something, you can go after this $5,000 prize.
Hey.
That's cool.
Yeah.
Was last year's video about funding science, or was it a different topic?
No, it was basically the same kind of thing, and it had a slightly different title. Stand with Science is what it was last year.
Okay.
Next year it'll be Stand on Science.
Well, and it's interesting because last year's, you can see in the beginning of the YouTube video the hand of the guy drawing the graph, and he draws the graph, and then he says, and here's what would happen if the sequester goes into effect. So,
Cool.
and then he says, and here's if the sequester cuts are permanent, here's what will happen.
Right.
It's an impressive video, so that alone is worth checking out.
Cool.
I'm sure they get lots of submissions.
Good.
Yeah, that's cool. We have a listener pick from Peter who writes, Dear TWiV team, I was sent this and thought that it may make a listener pick of the week. A 12-point checklist for assessing the validity of health stories in the media. Statisticians Kevin McConway and David Spiegelhalter, writing in Significance, a quarterly magazine published by the Royal Statistical Society, oh my God, have come up with a checklist for scoring media stories about medical risks. They have called their 12-item mnemonic checklist the John Humphreys scale. John Humphreys is a well-known UK radio and television presenter. And the checklists are just observing people, original information unavailable, headline exaggerated, no independent comment, higher risk, unjustified advice, might be explained by something else, public relations, puff, half the picture, relevance unclear, yet another single study, small. To use the checklist, score a point for every yes answer. Here, yes is bad, and points mean poor performance. Score a point also if you cannot tell what your answer should be, because not providing relevant information should generally ring as many alarm bells as providing adverse information. Count up the points. The nearer to the score is to 12, the less notice you should take of the story. That's very good. Hey, how would TWiV do here?
We'd get a 0.
It's right.
That's right. That's exactly right.
TWiV is found at twiv.tv and on iTunes. And over on iTunes, there are lots of podcasts. And we are generally on the front page of the Medicine and Science podcast directory. And that's because a lot of people make comments and leave ratings for us. So if you want to help us, go over there on the iTunes directory and do that. We really appreciate being more visible. And do send us your questions and comments to TWiV at twiv.tv. Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thanks. This was fun. I learned a lot.
Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
Quite welcome. Always a good time.
Alan Dove is at turbidplaque.com and also on Twitter. Thank you, Alan.
Always a pleasure.
And Dickson Despommier is at verticalfarm.com, medicalecology.com, and Racaniello.org, and somewhere in the Huffington Post empire.
There you go.
Thank you, Dickson.
At the bottom of that list, unfortunately, but that's okay. We're working on it.
Thank you, Dickson.
Had a great time.
Good to see you.
Yep, same here.
I am Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.